Okay, so all of this stuff was found in page 239. If you guys go to page 239, here it is, right here. A germ cell is a cell that's located in a part of an organism that produces your gametes. For humans, that would be cells that make up testes and ovaries that eventually will go through meiosis and produce sperm and egg. A somatic cell, think about a somatic cell as every single cell in the body that's not part of sexual reproduction, otherwise testes and ovaries. So your skin cell, a brain cell, a liver cell, a blood cell, a bone cell. These are somatic cells. But the question asks you, which type can be passed on to future generations? Which type of mutation? Well, if there's a mutation in a germ cell, then that's going to affect the sperm and egg, and that mutation may show up in your kid that you yourself don't have. If there's a mutation in some DNA on like a cell that makes up your arm, like the skin on your arm, your kid's not going to receive that mutation. So only the germ cell mutations can be passed on. A lethal mutation right here, right, can cause death, often before birth. Some mutations, however, result in phenotypes that are beneficial. So some mutations are actually good for us, which kind of leads us to the second part, right? How are mutations important in evolution? They create what we call variation. If there were no mutations ever, then all life on this earth would have been a bacteria cell, and it would have stayed as a bacteria cell. It just would have copied and copied and copied and copied itself. It is mutations that brings out new traits that never before existed. And so we, human beings, and every other different species on the planet are products of billions of years of genetic mutations that were for the better, that increased our fitness. Okay, so we also had to define the following terms. Chromosome mutation. It's going to be the Chromosome mutations involve changes in the structure of a chromosome or the loss or gain of a chromosome. Deletion is the loss of a piece of a chromosome. Inversion is a chromosome segment that breaks off, flips around backwards, and reattaches. And a translocation is a piece of one chromosome that breaks off and reattaches to a non-homologous chromosome. That would be like a piece of chromosome number 21 breaking off and attaching to a piece of chromosome number 15, which is another way or another type of Down syndrome. Nondisjunction is where you have your homologous chromosomes. You have your homologous chromosomes. So there's like, there's dad's chromosome number 1, and there's mom's chromosome number 1, and they're supposed to split apart where one gamete has one chromosome and the other gamete has the other chromosome. That's what should happen in meiosis. Nondisjunction is when these guys, which get together really closely in prophase 1, they get together really closely, and a lot of the times what you get is this crossing over that occurs, right? And they kind of overlap. They get stuck together, and both of them end up going into one gamete. And so if a sperm were to come along, right, which has its haploid, so it only has one copy of each set of chromosomes. If it were to fertilize this, you end up getting trisomy, right? Where you end up getting that 3 chromosomes together. This one would have 3 chromosomes. If it fertilizes this egg, you end up only having one of the 2 copies. You end up having monosomy. If this is chromosome number 21, you have trisomy 21, and that is another name for Down's syndrome. Right? And if you look at the diagram in your book, you can see this individual's karyotype, which is a picture of all your chromosomes, has 3 chromosomes right there, and specifically chromosome number 21. You guys were supposed to go to this link up here, right? And then you're supposed to see what does the chromosome lengthen or shorten, and what is the prognosis? What happens when you have a duplication, deletion, inversion, or translocation? So that looked like, by the way, this is nondisjunction right here. This is what happens. It can either happen in the first part of meiosis, meiosis 1, where they stick together and you get trisomy here and monosomy here. Or they can separate fine in meiosis 1, but then the sister chromatids don't separate in meiosis 2, and you still end up getting screwed up gametes, trisomy here and monosomy here. It's just only in half of the gametes. These guys are going to be fine. So back to the Webquest, right? So we're in this part 2 of the Webquest. It wants you to know, well, first of all, in duplication, do we lengthen or shorten the chromosome? Well, we're duplicating it, so it's longer. The prognosis is duplication of the whole genes is not usually serious. In fact, copies of genes may provide the source material for new genes to evolve. You just have more of what you already have, so it's typically not that big of a deal. Deletion, however, we're shortening the chromosome because we're deleting whatever is in this big chunk of a chromosome. So all the genes that used to be here are gone now. Most gene deletions result in a miscarriage, so that's the prognosis of the embryo, or produce serious and usually fatal abnormalities in babies who are born alive. Now, an inversion is where this little piece right here, right, this piece right here, breaks off, flips upside down, and reattaches. So you have this kind of inversion, this flip around. And so does it lengthen or shorten? It does not either. It stays the same length. You just have a flip around. Inversions within the chromosome are usually also not serious because all the genes are still present. And then translocation. There's two types of translocation. A simple translocation is where a piece of one chromosome, let's say chromosome number 1, detaches and attaches to another non-homologous chromosome. So it like attaches to chromosome number 14 or chromosome number 17. That's not supposed to have those genes. What does this do in terms of shortening or lengthening? It shortens this one, but what does it do to this one? It lengthens the other. One form of Down syndrome is caused by a fragment of chromosome number 21 being attached to chromosome number 15. So it can cause some abnormalities. And so that's the answers to all of part 2. There we go, sir. Okay, now we're on part 3, and you're supposed to go to this quiz right here, okay, and watch this video, right? It talks about all what happens with all 4 chromosomes, and you answer the 5 questions. An extra copy of a chromosome is a duplication, so you're supposed to write that down for number 1. When you have something breaking apart, flipping around, that's called an inversion. A piece of chromosome breaks off and is attached to another chromosome. That is a simple translocation. A reciprocal translocation is where one chromosome breaks off a piece and puts it on another chromosome, and that chromosome breaks off a piece of itself and puts it on the other chromosome. So you have 2 different chromosomes swapping parts of their DNA. Number 4 is true. Yes, chromosome mutation is a substantial change in the structure of a chromosome. And both inversions and translocations can lead to overall deficiencies, which means something is wrong or bad, and duplications of genetic material. That is also true. So all of those answers should have gone right here. You just wrote all those out. The last one is kind of, or the last 2. In general, chromosomal mutations usually have much more serious than single gene mutations. And the reason for that is a chromosome, right, is a long piece of DNA coiled up, right? Here's a long piece of DNA. Here's that little piece of DNA. One gene is just one section. So if you mutate one gene, you're only going to screw up the protein, right, that this translates for. But if you screw up a whole chunk of a chromosome, you're going to affect many genes. And obviously, that is going to be more serious. List 3 specific environmental agents that can cause chromosomal damage or mutation. UV radiation can really affect the chromosomes that are in your skin cells specifically, right? We know that smoking, right, has a lot of chemicals that can bond to your DNA and cause problems. And then also chemicals that we are exposed to not only in the air and our environment, but in our diet. Anything that causes our DNA to mutate, we call those mutagens. And that's never good.